Good morning, dear students. My name is Dr. Ravi Babu, Assistant Professor from Mallarity Institute of Medical Sciences, Department of Biochemistry. Today I'm going to teach you carbohydrates metabolism. Chapter name is in the subtopic glycolysis. So here, first of all, in glycolysis, we are going to directly glycolysis pathway. Another name is Embden-Meyerhof pathway. So, what are the subtopics? What are the important points in glycolysis we are going to cover in this? First of all, introduction and biomedical significance of glycolysis, types of glycolysis, aerobic, anaerobic, reactions of glycolysis, complete and reactions, clinical applications, energetics of aerobic and anaerobic glycolysis, a regulation of glycolytic pathway, glycolysis in RBC, that hypoxic conditions that is covered under Rappaport lowering pathway 2,3-BPG shunt, source and fate of pyruvate, PDH multi-enzyme complex, fate of acetyl-CoA, and importance of glucose 6-phosphate and glucose 6-phosphate snakes. What is glycolysis? That's Embden-Meyerhof pathway, glucose to pyruvate aerobic and anaerobic here glucose to lactate that provide energy and intermediate to other pathways. Glycols occur in 10 sequence steps catalyzed by enzyme that is glucose to pyruvate. So 10 sequence steps, enzymes. We'll see in detail. Glycols is a major pathway for regulation of glucose that occurs in cytosol, all cells. Here aerobic 10 ATP, anaerobic glycolysis 2 ATP. Insulin favors the glycolysis. Glucagon inhibits glycolysis. Glycolysis, there are phase of glycolysis, preparatory phase, payoff phase, or preparatory phase, glucose activation phase, and payoff phase to energy extraction phase. In 2 ATPs are invested and hexose cleaved to 2 triose phosphates in preparatory phase. And payoff phase are energy extraction phase, conversion of glyceraldehyde 3-phosphate to pyruvate and 2 ATP occurs. These steps after 5 will constitute. So reactions of glycolysis. Glucose irreversibly phosphorylated by ATP to form glucose 6-phosphate by hexokinase or glucokinase. So first rate limiting step and committed step is this, and glucose favored by insulin, hexokinase not. So glucokinase favors by insulin. Reaction to glucose 6-phosphate is isomerized to fructose 6-phosphate by the enzyme phosphohexose isomerase. It is reversible enzyme. Just see the glucose molecule in this glucose hexokinase or glucokinase, presence of ATP. After releasing 1-phosphate, they bind with 6 carbon of glucose. They form glucose 6-phosphate, and this is irreversible reaction. The next one is a reversible reaction, that is phosphohexose isomerase. Just changing the name from glucose to fructose. So phosphohexose isomerase. So instead of glucose, we see here fructose 6-phosphate. Reactions of glycolysis. Third one, fructose 6-phosphate is irreversibly phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase in presence of ATP, that is reversible. So second rate limiting, that's a committed step. Reaction 4, fructose 1,6-bisphosphate undergo aldol cleavage at C3C4. We're repeating this bond into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by aldolase enzyme, that is reversible. See now fructose 6-phosphate to fructose 1,6-bisphosphate, first carbon 6 carbon, one more ATP, they split into ADP with presence of magnesium ions. Enzyme is phosphofructokinase. So here aldol is a reversible reaction. The above one is irreversible. 3 carbon they split. So glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. Dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by phosphotriose isomerase, that glucose to 2 molecule of glyceraldehyde 3-phosphate. Reaction 6. Here 2 molecular glycerol 3-phosphate undergo phosphorylation and oxidodehydrogenation. So 2 molecular glycerol 3-phosphate undergo phosphorylation and oxidodehydrogenation. Now look here, glycerol 3-phosphate. Here they convert to 1,3-bis phosphoglycerate, glyceraldehyde 3-phosphate, dehydrogenase, reversible reaction. See now here reversible reaction. You'll get 1,3-bis phosphoglycerate. They go back, they form after releasing 1-phosphate, you'll get glyceraldehyde 3-phosphate. That's in gluconeogenesis. 1,3-bis phosphoglycerate. So again, what happened here? Phosphoglycerate kinase. So they release one phosphate group from position number 1 carbon. Then you will get 3-phosphoglycerate. Presence of magnesium ions split again here ADP to ATP biosynthesis here. So they take one phosphate from first carbon. You will get 3-phosphoglycerate. So phosphoglycerate mutase, that enzyme, they change position number 3 to 2 carbon, that is 2-phosphoglycerate. Second position 2-phosphoglycerate. So enolase enzyme, they change 2-phosphoglycerate to phosphoenol pyruvate. So here we'll get phosphoenol pyruvate, just changing position number. So in this position here we'll get keto group. So enolase. So double bonds are there. So phosphoenol pyruvate. So ADP to ATP, again one more substrate level phosphorylation, pyruvate kinase, you get pyruvate. Okay, so C carboxylic group and CHOH, CH3. So just because of this, still they continue enol group. So double bond is there. So now we look here. Overall, glucose to glucose 6, fructose 6 phosphate, 1,6-bis phosphate, aldolase, glycerol 3 phosphate, isomerase is the same thing. Now look, this one we discuss from 1,3-bis phosphoglycerate to 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. Next spontaneous changes, they form pyruvate keto group. So enol group to keto group. But here, once they go to in anaerobic glycolysis, lactate dehydrogenase. Presence of lactate dehydrogenase, you'll get lactate. So from this coupled reaction, here number, here 10th reaction, so 5th one, just remember here whatever we generate NADH plus H plus, that's utilized here to change pyruvic acid to lactate. So NADH plus H plus, they when they bind with these hydrogen atoms, so very important, they bind with fire, they form lactate. So again dehydrogenation reaction, NAD plus is there, again regeneration, they continue this pathway. So 2-phosphate, same thing continues, 2-phosphoglycerate, phosphoenol pyruvate to change same reaction. And one more important, anaerobic glycolysis. So energy production anaerobic glycolysis. So here glucose plus ATP, glucose 6-phosphate, ATP is used. Reaction number 3, fructose 6-phosphate plus ATP, you'll get fructose 1,6-bis phosphate, one more ATP used. So 2 ATP utilized here. Reaction number 7, 2 molecule of 1,3-bis phosphoglycerate, 2 molecule, so we'll get 2 molecule of 3-phosphoglycerate. So substrate level phosphorylation, this is the ATP substrate level phosphorylation, ATP with size. And reaction number 10, 2 molecule of phosphoenol pyruvate to 2 molecule of pyruvate, again 2 more ATP. And here, so comparison of glucokinase and hexokinase, how we compare? So here site present only in liver, that is glucokinase. Hexokinase present in all tissue including liver. So activity depends on nutritional state of glucose induced and active at high blood glucose level, that is glucokinase, but does not depend on the nutritional state of glucose, that is hexokinase. Function active only when blood glucose levels are high, hyperglycemia, so favors glycogenesis, that's important, glucokinase. But here active, this enzyme is active even at low blood glucose level for the utilization of glucose for energy. So Km value, high Km value, low affinity to glucose of glucokinase, low Km value, high affinity to glucokinase of hexokinase. Induced by insulin, glucokinase not induced by insulin. So inhibition, no direct feedback inhibition by glucose 6-phosphate, but hexokinase feedback inhibition by glucose 6-phosphate. So what is Rappaport lowering cycle? See now within source and fate of pyruvic acid before that. See how you get from malate to pyruvate, malic enzyme, glucose to glycolysis also will get pyruvate, and so reversible reaction of lactate to pyruvate and alanine to pyruvate. And this pyruvate enter into mitochondria like with presence of pyruvate carboxylase will get oxaloacetate. Oxalo, oxaloacetate will get oxaloacetate. So here pyruvate dehydrogenase complex. So we'll get with help of this one acetyl CoA. So the source of pyruvate alanine, glucose, malate, lactate 1, 2, 3, 4. Glucose glycolysis, malic enzyme, LDH, ALT, alanine aminotransferase also will get pyruvate. So since of oxalation from pyruvate and acetyl CoA also from pyruvate. So what do you know about pyruvate dehydrogenase complex? So simple thing, pyruvate to acetyl CoA, there are 5 coenzymes, 3 main enzymes. So now see now, here pyruvate inactive, pyruvate active, when they are active. So if dephosphorated state that is active. So active to inactive by phosphorylation state, then inactivation prompted by high ratio of acetyl-CoA and coenzyme A, NADH, NAD, ATP, ADP. So high, if they are high, inactivation is promoted. Activation is promoted by pyruvate, NAD plus coenzyme A, any sudden demands upon the cell signaling by calcium ions. Once they release phosphate group, see now PDH phosphatase, they release phosphate group from phospholate pyruvate will get active. Then only they change pyruvate to acetyl CoA. So this is the pyruvate. What is PDH? So here 1, 2, 3 important enzymes. What are these? First of all, pyruvate decarboxylase, dihydrolipo, amide acetyl transferase, dihydrolipoyl dehydrogenase. So important pyruvate to carbon dioxide. Here one, so from thymine pyrophosphate, this is a one coenzyme, and second this is FAD to FAD, B1, B2, B3. So B1 from thymine pyrophosphate, B2 from FAD, and B3 niacin from NAD. Lipoic acid is there, and from coenzyme A from pantothenic acid. So pyruvate to carbon dioxide, this is pyruvate decarboxylase TPP to hydroxyethyl TPP recording. So TPP to hydroxyethyl TPP pyruvate decarboxylase. So release of CO2 will get hydroxyethyl TPP. So here this hydroxyethyl TPP, when they bind with oxidized lipoic acid, they change to acetyl lipomide. This acetyl lipomide, when they bind with coenzyme A, they form acetyl CoA. This is of the product. And reduced lipoic acid, again they bind with FAD, they form FADH2. So presence of enzyme dihydrolipoyl dehydrogenase. This FADH2, it leads to synthesize NADH plus H plus when they bind with NAD. So overall fate of acetyl CoA. So what happened this fate of acetyl CoA? Here carbohydrates, protein, lipids, we get ultimate product that is acetyl CoA. This acetyl CoA is utilized to synthesize fatty acid synthesis, post-translational modification of acetylation as a co-substrate in drug metabolism. So as a co-substrate. And this acetyl-CoA utilized to synthesize ketone bodies, synthesis of neurotransmitter acetylcholine, synthesis of melatonin. So synthesis of melatonin, synthesis of neurotransmitter acetylcholine, synthesis of ketone bodies. And this acetyl-CoA utilized to and they enter into TCA cycle and synthesis of cholesterol by synthesis of steroid hormone. So how many, what is the utilization of it? Again, overall, fate of acetyl-CoA. Here, fatty acid synthesis, modification, post-translational modification, like acetylation as a co-substrate for drug metabolism, and synthesis of ketone bodies and neurotransmitter acetylcholine, melatonin synthesis enters TCA cycle for generation of reducing equivalents to generate ATP with help of electron transport chain, synthesis of cholesterol, and synthesis of steroid hormone. So we get this acetyl CoA from carbohydrates like glycolysis, we'll get after glycolysis with the help of link reaction. What you get? So we'll get acetyl-CoA that enter into TCA cycle. And from protein, so glucogenic amino acids, lipids here, for example, if you think we take about beta oxidation of fatty acids, there you'll get acetyl-CoA. So based on that, for example, if you take palmitic acid, you'll get 106 ATP. Same, they enter into TCA cycle 8 times, so 80 ATP they produce. So key reaction of gluconeogenesis, we'll discuss this later. Thank you.